Okay, you can carry.
Okay, so again, very good evening, everyone. So my name is Abhishek Presta, and I'm a marine biologist working in National Institute of Oceanography, like Chetan has said. I work mostly on plankton and microbiology, as well as the molecular aspect of these seawater microbes. So today I will be talking about a very familiar topic. Uh, that is on zooplankton. Mostly, I'll be focusing on plankton and not the zooplankton alone, but I'll try to make things simple and convenient for everyone to understand. So, is my slide visible?
Yes, it's visible. Yeah, it's visible, Abhishek. You
Okay.
can carry on.
Okay. So, the topic that I'm going to discuss today is zooplankton, an important component of the aquatic ecosystem. So, introduction. So I'll start from the very basic thing, that is the aquatic ecosystem, which comprise mostly of water. As you all know, water is a very important component for any life form that is living in this planet, and it is a very important factor for the survival of every individual species. An organism can survive without food for a while, but without water, it's very difficult to sustain life. As we all know, 70% of our planet comprises of water, which consists of lake water, fresh water, marine water, the tributaries, ponds, stagnant water, all this together. And these water bodies, they are being harnessed by natural processes or human intervention in the form of, say, natural process such as withering of soil, erosion, then you have runoffs through rains and all these natural phenomena. Whereas human intervention mostly refers to the use of fertilization, which gets washed off to the water body, nearby water body, and ultimately to the oceans that hampers the overall marine life. In order to assess the degree of this what intervention, be it human or natural, various assessments are required, and these assessments can be categorized into physical, chemical, and biological parameters. So, for most of the biological parameters that are being assessed is mostly in the form of indicators. That is, certain organism living in a particular ecosystem gives the idea of the water body, whether it is harmful or polluted or any other contaminant present. And one such organism is plankton. And planktons, mostly phytoplanktons, which serves as an ecological indicator. One such example can be given as Canidesmus species, which generally gives an indication that the water is polluted. So let's come to the main topic for the day, that is plankton. Now, plankton in general is a diverse group of microscopic living organism that drifts around a water body. They do not move on their own. They do not have any locomotory organs in the body. They simply drift through the movement of water currents and flow of water. And plankton is a collective term. It doesn't refer to a particular individual species or organism. So it includes mostly diatoms, dinoflagellates, desmids, larval forms, algae such as blue green algae and other algae, even bacteria, some jellyfishes, protists, rotifers, and other smaller protozoans and metazoans. Now, the singular term for plankton is called plankton, which people don't usually use because it's very complicated, as plankton itself is a meaning in a plural form and refers to a various collective group of organisms. The term plankton was coined by Victor Hansen in the year 1887, which is mostly he's mostly an ecologist who works on freshwater planktons and other marine planktons as well. So the general size of plankton is from 20 micrometer to 2 centimeter. So there are exceptions where the plankton is less than 20 micron or more than 2 centimeter, but they fall under different categories. So the base definition or the size for a plankton is 20 micron to 2 centimeter. They are found in freshwater, estuaries, or marine environment, depending on their ability to adapt in the particular environment. Broadly, planktons are classified as phytoplankton and zooplankton. Phytoplankton being the or plant type, and zooplankton being the animal type, which includes mostly larvae and other smaller metazoans and protozoa. So, the planktons are unique in the marine environment because of a very particular ability, that is to fix inorganic carbon and to build up organic matter through primary production. Uh, and that is why the study of plankton is of prime importance. Study of plankton in this case is mostly for phytoplankton because they have chlorophyll, which is the very important pigment used for the process of photosynthesis. As you all know, in the presence of light, making carbohydrates and releasing oxygen as well. Based on the size of phytoplankton in this case, we have picoplanktons, ultraplanktons, nanoplanktons, micro or the net size plankton, and macroplanktons. Apart from that, we have zooplanktons, which are more larger in size, that is more than 500 micrometers. So planktons contribute to about half of the photosynthesis of the planet, making them one of the most important producers of oxygen. So trees are not the only organisms or only life form which gives oxygen to us. Even phytoplankton contributes equally, or the algal part of the phytoplankton, which contributes to the production of oxygen. They also make simple chemicals because they contain chlorophyll and live in the upper zone to get sunlight, of course, because they need to undergo photosynthesis. And phytoplanktons are in turn being eaten by swarm of other plankton called as zooplankton. So they are a very important aspect in the food chain because they are being eaten. Phytoplankton are being eaten by zooplankton. Zooplanktons are being eaten by other larger zooplankton. Those are being eaten by fishes, and similarly the uh, chain continues. So here are a few examples, a few pictures of zooplankton that I have observed under the microscope. These are the pictures taken by me. Cyclops, Bospina, Nauplius larva, Caratella, Polyarthra, and Diphorodon. These are all freshwater plankton and found mostly in stagnant water such as ponds or stagnant lakes. Next is a plankton slide, uh, group of plankton which is being counted on a slide. You can see on the left corner, and other slide shows the zooplankton, which has the larval forms as well as the other zooplankton organisms present there. You have trophophore larva, you have the lobster larva, the crab larva. All those larval forms are present. These slides shown in the picture are mostly marine species, and I found the estuarine and marine environment. Uh, so Nauplius larva here in this case, I will show you here. This Nauplius larva, it is common for zooplankton, for most of the zooplanktons in terrestrial as well as marine environment. In terrestrial environment, Nauplius larvae, they develop to form other freshwater insects or insect species, and in the marine environment, Nauplius larvae forms various groups such as crustaceans, barnacles, or other groups of molluscans as well. So the Nauplius larva is a very great significance, or more you can say a higher chances of forming other developing to other organisms. So next is we have some other picture. This is the first one you can see is the lobster larvae, mm, the stomated lobster larva, and below that we have the jellyfish larvae, and after that we have copepod larvae in the right corner. You can see copepods are also very important group in the zooplankton family. They constitute majority of the zooplankton family, and they are of 10 species, out of which three of them are very common and free living, found in the what you call aquatic system. And the picture here you can see is a cyclopod copepod larva, which is the most common of all the copepod species. Apart from that, you can see three other pictures, that is Oscillatoria, Storostrum, and Peristrium. If I think those are clear enough. These are, excuse me, these are freshwater phytoplanktons, and they mostly contribute to the chlorophyll distribution and acts as food for other zooplankton. Oscillatoria is a major phytoplankton algal group present in the what you call stagnant water bodies mostly. So let's come to the materials that are required for collecting planktons. Firstly, we have plankton net, which are very big, huge net used for collecting plankton, for filtering plankton. And then you have the plankton collecting tube, which is attached at the end of the plankton net for all the plankton which are filtered through the net to be collected. Then we have preservatives, which 10% formalin used mostly for zooplanktons, and Lugol's iodine, which is used only for phytoplankton. Then you have the sediment after chamber, which I think you have seen in the previous slide. It's a special kind of a slide which is used for counting cells. And here in this case, these are rectangular boxes where you can count this zooplankton or zooplankton mostly for quantitative study. Then we have deep well plates. Those are 6 12 wells plates. You can see picture in the other slides. And finally, microscope. Here you can see a picture, general structure of a plankton net. Now, a plankton net, you can see the net, the structure is very conical in shape. And at the end of the net, you have the net bucket, which is the plankton collecting tube, where the filtered plankton are collected. The net is mostly made up of nylon or dacron mesh, and the mesh size generally ranges from 20 micron to 50 micron. Then the net is being attached with a net ring for holding to a cable wire with the help of bridges, and this cable wire is being attached to a boat for dragging or for towing at different depths. Apart from that, to measure quantitative zooplankton specimens, you can simply filter water a certain amount to a definite proportion to get the desired plankton. And apart from that, next we have the deep well plates. Here you can see you have 6 wells, 4 wells, 12, and 24 wells plate. These plates are used for counting phytoplankton mostly, and phytoplankton which are alive. You can also see them and then culture them. These plates, apart from counting, they are also used for culturing of phytoplankton group, which at present is a very uh, progressive work, you can say. Then below that you have two other pictures of schedule chapter. One is the original picture of how it looks, the slide, and there's a diagrammatic representation. You have a trench in the middle with square boxes where you pour your sample. Generally, the size that goes there is 1 ml, so you can take 1 ml of sample at a time, cover it with a cover slip, and count it under the microscope. Stereo microscope will be sufficient. So let's come to the methodology of how these planktons are collected. First is, like I said, quantification by filtering large volume concentrated to its respective proportion. So what you can do is take the net, filter a certain amount of water, say 50 liters of water, and you concentrate that to 5 ml. So concentrating 50 liters of water to 5 ml gives you a definite proportion filtering, filtered uh, plankton sample. Then apart from that, you have the plankton net dragged through the water behind the trawler. So like the previous image, you have seen the structural figure of a net. The cable is being attached at the end of a ship or a trawler, and the trawler is being dragged in the ocean from one part to another part, and the zooplankton or the phytoplankton which is being collected in the tube below is filtered through the method of dragging. Apart from that, we have another method, that is by dropping the net at a certain depth. So what you do is you drop the net, like you can see the picture in the slide. You drop the net at your desired depth, tow it for a while around the area, and then you slowly pull it back up for the samples to be taken. Apart from that, a simple method of phytoplankton preservation is by taking the water sample directly from the source and fixing it with Lugo lighting. After that, siphoning it through a tube and then counting it in an instrument which is known as flow cam. So that will give the amount or the number of phytoplankton group that is being present in the water. So apart from that, now here are some physical parameters that we will be talking about. These parameters also influence the growth and the overall productivity of plankton in an environment. So these parameters are equally important and are very much correlated to the overall biological parameters that we have been discussing about. So starting with temperature. Temperature plays a very important role in the growth and productivity of your phytoplankton as well as zooplankton. So temperature is generally measured using thermometer, and the temperature, higher or lower temperature, generally determines the uh, overall growth of a particular larva or maybe a phytoplankton group, because the temperature for this what do you call, for their processes are also important to carry on in the next step. Excuse me. Next is we have turbidity. It's also called transparency. That is like you can say how much the amount of sediment that is being present in the water body from where the water sample is collected. Now, turbidity is generally measured using a particular device called as the Secchi disk. Now, Secchi disk it is being dropped in the water body and measured the length of the depth at which the Secchi disk is completely visible. So the length or the depth gives the degree of turbidity of the particular water body. A higher turbidity generally reflects that the water body has comparatively less phytoplankton groups and a higher number of zooplankton, because the coloration also plays an important role and it hampers the movement of the phytoplankton. Then you have rainfall. Of all the parameters, rainfall is a very important parameter because it completely changes the dynamics of the water body. Since rainfall brings runoff from different areas, the overall system of the water body changes. The phytoplankton number, the phytoplankton quantity, the phytoplankton productivity, zooplankton numbers, as well as the nutrient composition, everything shifts because of rainfall. Higher rainfall generally lessens the number of phytoplankton in the area and also affects the overall dynamics. Apart from that, we have salinity and specific conductivity. These two I have kept it together because of one reason. Now, salinity is the measure of salt contents, and it is only seen in marine or estuarine environment and not freshwater ecosystem. So for freshwater ecosystem, salinity will be zero. But for estuarine and saltwater, fresh marine water, we have a certain degree of salinity. Whereas specific conductivity is the measure of ions or minerals present in the solution in the water body. So specific conductivity is particularly for a freshwater ecosystem. You can measure specific conductivity in marine water and ecosystem also, but since you are measuring salinity, the mineral ions are already being considered there, and thus specific conductivity doesn't hold much importance here. Salinity can be measured using a refractometer. Salinity mostly ranges from 30 to 35 in a marine ecosystem, and 5 to you can say around 30 is for most estuarine areas. Specific conductivity, on the other hand, is used with an instrument present in the laboratory, which is known as specific conductor, and it gives a very definite value for the number of mineral ions present in the water. Another factor is chlorophyll A. The chlorophyll A is a very broad term or broad thing you can say for phytoplankton mostly and not zooplankton. So the higher value in chlorophyll will directly reflect to the high number of phytoplankton in the particular water body. Uh, chlorophyll levels generally gives a direct relation to phytoplankton and not zooplankton. Chlorophyll can be measured using various parameters. Firstly, by filtering the water through filter papers and keeping the filter paper in acetone overnight, and then measuring the absorbance in a spectrophotometer. And the degrees, the values are being calculated further, and the chlorophyll values are given. Generally, chlorophyll for um, estuarine or marine environment ranges from 5 to 10 mg per meter cube. Sorry. Then next term you can see is SPM. SPM stands for suspended particulate matter. As the name suggests, suspended particulate matter are those particulate matters which are present in the water body. High level of SPM will generally link to high turbidity, and lower SPM will result to lower turbidity. So we are measuring SPM after turbidity because we need to know the value of how much suspended particulate matter is present in that water body, because it is directly linked to the movement of the plankton groups, so it causes hindrance to in the movement, and might also result in the certain uh, decrease in the phytoplankton group. So SPM is also measured same way like chlorophyll through filtering, but in this case for SPM, we use 25 filter paper for measuring SPM, which is ashed, or you can say preheated overnight, or not overnight, at 450 for 3 hours, and then cooled overnight and used it the next day. The initial weight and the final weight after and before filtration and after filtration gives the value of your SPM. Next is TEP. TEP is a very complicated parameter. It stands for transparent exopolysaccharide. Now, TEP, since it's quite complicated, I won't go in details. I'll just explain in a very simple manner. Now, TEP, what will happen is in an environment, phytoplankton as well as bacteria, they undergo certain mechanism, and in that process, they release certain substances in the water body. One of the substances is dissolved carbohydrate. Now, this dissolved carbohydrate is being released by phytoplankton as well as bacteria. So, since phytoplankton are larger in number, the dissolved carbohydrate are more in number because of that. And since the dissolved carbohydrate are, carbohydrate being sticky in nature, it attracts other debris which is surrounding the environment, and after attracting, it sticks with the carbohydrate molecule, and gradually it sinks down. Now, TP is also important because it helps in the sinking carbon footprinting and other certain processes that's very common in nature. It contributes to that as well, and the major contributor of TP is phytoplankton and bacteria. So TP, like SPM and chlorophyll, are same way measured through filtration. But in case of TP, the filter paper used is isocarbonyl 0.45 filter paper, special one, which traps the dissolved carbohydrate. And this filter paper is further processed using a stain called Alcian blue and 80% H2SO4, and this is being analyzed under spectrophotometer, and the absorbance after further calculation gives the value of your TEP, that is the transparent exopolysaccharide. After that, we come into the chemical parameters. That is, first is a very common one, the pH. You can see the degree of the negative logarithm, hydrogen ion logarithm, the amount of hydrogen ion that is being present. So pH is generally a measure for determining whether a water body is alkaline, neutral, or acidic. So acidic water mostly seen in the fresh water where the range is acidic to basic, whereas ash-rain and salt water are always have an alkaline pH. Dissolved oxygen is a very important parameter for phytoplankton and zooplankton and other organisms living in the aquatic ecosystem as well. So dissolved oxygen, simple enough, and I think most of us have heard about it. And to what you call it, determine the value of dissolved oxygen, we use Winkler's method. That is two solutions of Winkler A and B, manganese sulfate and alkaline iodide, which is being fixed using H2SO4 and further titrated using sodium thiosulfate. Next is BOD. BOD, biological oxygen demand. Now, biological oxygen demand is the amount of oxygen the water body demands. Now, one thing to note here is dissolved oxygen and BOD are completely opposite. They are measured the same way, but they are completely opposite. In an environment, if the dissolved oxygen is higher, there are chances that the BOD will be less, and vice versa. So in case of an environment or an ecosystem where eutrophication happens, such as algal blooms or large growth of algae in a water body, the amount of dissolved oxygen will be less, but the UOD will be very high. So that relates to the phytoplankton that is there in the environment. Next is the chemical oxygen demand, the COD. Chemical oxygen demand is mostly for polluted water. It is the measure through which you can determine the number of oxygen required to disintegrate or react with the pollutants present in the water body. It is mostly for water bodies where high contaminants or sewage influence or such polluted waters are present, where the phytoplankton numbers are quite less and zooplankton's number are almost nil or considerably very low. Apart from that, we have other chemical parameters, one of which is free carbon dioxide. Free carbon dioxide is a measure of the amount of carbon dioxide molecule present in the what you call free state. So it is generally measured like same way like titration, where we use phenolphthalein as an indicator and titrate it using sodium bicarbonate. Apart from that, we have total alkalinity. Total alkalinity gives the amount of or the measure of the degree of alkalinity in a water body. This is also same way used phenolphthalein as an indicator as a first indicator, and after that we use methyl orange as a second indicator for during titration, and the titrant uses standard H2SO4 solution. For total hardness is the hardness. Hardness generally refers to the calcium ions or bicarbonates which are being present in the water body in the form of ions. These are generally determined the same way like using titration method, and titration are generally done using EDTA, and the indicator uses erichrome black tea and starch. Uh, Apart from the ones we have discussed, coming to the next very important parameter, that is the nutrients. Nutrients are very important because the intake of nutrients by phytoplankton helps in the process of their growth, and which further goes in the food web and circulates around. The nutrients that are being considered in this case are mostly nitrate, nitrites, silicates, and magnesium, uh, sulfur, and phosphorus. These few nutrients are being used for, I mean, considered for this. So chemicals and physical and chemical parameters that we have discussed so far are very important because they affect the overall growth and living of the phytoplankton and zooplankton. Coming to few applications of plankton, zooplankton mostly, and phytoplankton as well. So they serve as food for many larger animals, and which indirectly helps us humans as well. Since most of the zooplanktons are used as food by many organisms living in the freshwater or marine environment, which we humans consume. So they are very important because they act as food for our food. You can say. Next is another important application of plankton is in the fishery and the aquaculture industry. Phytoplankton and zooplankton are being used as feed for various fishes which are being cultured in the aquaculture farms and ponds. So they are very important and uh, helps in the growth of the fishes and provides the necessary nutrients for the fishes as well. Then you have large scale cultures is technically feasible. Large scale cultures for particularly for phytoplanktons in this case, because phytoplanktons, as I have said earlier, they have at present culturing of phytoplankton is very much culturing of phytoplankton is very much prominent, and they are being used highly in most of the researchers these days. And these cultures are very important in the sense that high content proteins are being derived because large scale production of phytoplankton will generally give more protein content, and because of that this protein contents which is usually more than 50% can be used for other processes for humans as well, because the amino acid constituent of the protein group in this case are very compatible and are very much used as well. So culturing phytoplankton uh, in a in a large scale in big industries or processes are very much common and are very much in demand these days because of its easy cultural techniques. Then you have another thing very this component is a very ongoing and hot topic these days. Phytoplankton may become increasingly important in space travel as a source of food and for gas exchange. Now, space going to space requires oxygen for the astronaut to sustain, and phytoplankton they are trying to use phytoplankton for this as firstly because protein content will be higher, so they can use it as food, and secondly using them as gases exchange, since phytoplanktons are plant like they release oxygen and that can be used by astronauts in their ships. Carbon dioxide released during spacecraft during respiration of spacecraft personnel would be transferred into organic substance by the algae, while the oxygen depleted during the process would support human life. Simple terms, they will help each other to sustain better and explore the space and our universe in a better way. So these are some importance. Apart from that, phytoplankton biomass in the oceans falls and rise according to multi-day cycles and appears to be sensitive in changes in surface area temperature, climate change, and ocean acidification. So you can say that phytoplankton is also contributes to climate change, and their biomass increase or decrease in biomass can cause very much uh, decline or the changes in the water quality, temperature, and other water parameters. Apart from that, another thing that is very important zooplankton group, which is known as the Antarctic krill, they are being present in the southern ocean, and they are being fed by whales in large quantity. So, the biomass is also important as the quantity of which the krills are being consumed by whales. Definitely, in the high productivity definitely keeps the balance, and thus phytoplankton and zooplankton are equally important as they keeps a balance for the organism above them, the food chain. So, coming to the conclusion part of today's and first is an adequate supply of nutrients, including carbon dioxide, enables phytoplankton and benthic algae to transform the light energy of the sun into energy-rich chemical components through photosynthesis. So, adequate supply of nutrients. Nutrients are already present in the water body. The other sources that through which nutrients are being increased or nutrients values increase or decrease in the water is through rainfall or by other external influence, human intervention. So, increase like for example, if you have a lake where the nutrient suddenly increases, you will get a excuse me, you'll get a high value of high number of phytoplankton because of high nutrient, and that lake will have a considered a bloom because of which the carbon dioxide will be less and the oxygen will be high. So apart from that, the light energy that is being provided by the sun through photosynthesis are also used to used by them. Uh, Apart from the next is bottom dwelling algae are responsible for about 2% of the primary production in the ocean, and remaining 90% is attributable to phytoplankton. So 98% of the primary production that occurs are mostly due to phytoplankton group, and the bottom dwelling algae are not much doesn't play much role in the productivity. Then we have most phytoplankton serves as food for zooplankton, but some is carried below the light zone. Since they go below the light zone, they not able to photosynthesize, as a result of which they die, and After death, the phytoplankton undergoes chemical mineralization, bacterial breakdown, or transformation into sediment. So, apart from serving as in the food chain, it also contributes in the environment through mineralization and other important processes that occurs in the water body. Phytoplankton production usually is greatest from 5 to 10 meters. So, within 5 to 10 meters, this is mostly for open oceans and water which are deeper, 5 to 10 meters or 6 to 33 feet below the this thing. Phytoplankton production is not much there, and first 5 to 10 meters usually has the high number because of it being a photic zone where light enters the water. So thus, we can conclude that phytoplankton are very important for the aquatic ecosystem as it serves as the basic food, source of zooplankton, which zooplankton being a connecting link between the arthropods and the heteropods. So thus, considering plankton to be a very important component of the ecosystem and serving as the connecting link between lower organisms and higher organisms and keeping a balance in the overall food chain, ultimately having a balanced food web as well. So, thank you.
Thank you, Abhijit. Now we are open for questions. You guys, students, especially if you have any doubts, you can get them clarified from Mr. Abhijit.
So, any questions, anyone? Feel free to ask. I may not be an expert in the field, but I'll try to answer all your questions with whatever I can.
You can post your questions in the chat box if you're not able to uh, on your mics. I will be asking the questions to Mr. Abhijit.
No questions here?
Okay, I think students have very much understood everything what you have told, Mr. Abhijit. Le- uh, so can you tell the students more about the different larvae that you find? Because some of like especially our zoology students have to study larvae and, later. So can you just expand on that if possible?
Okay, so larvae, there are forms. First, you have the larval forms, which as an adult, they decide to stay larval in the larval stage. And apart from that, you have the other larval forms, which, as I've said, grows into an adult, be it for crustaceans or other groups of organisms. So larval form, we have many larval form. Nauplius larval form, which grows to become cyprid larva, which ultimately becomes barnacle. So, excuse me. Barnacle, if you don't know, these are marine organisms which are found in the coast of a environment, and they are mostly biofoulos. You can say they since Nauplius larva can move on its own, not so much, but just a slight movement using its antenna present on the top. They can go around places, and once they develop to the cyprid larva, they move around in search of a place where they can settle. Since barnacles are a sessile organism, they cannot move. once they are their adult form, I mean, they need to stay in one place. So the cyprid larva searches for a particular place where it decides, or you can say it senses, and after sensing they decide to settle in a particular place, and in that place the larva, the antenna of the larva attaches to the floor, and after that it develops into an adult. Apart from that, Nauplius also developed to zoea larva. The zoea larva has three other forms: first, second, third form. Then zoea larva further develops into megalopa larva, and this develops to different groups of crabs that are being found in the intertidal region. So this is one. Apart from that, they also develop to lobster larva and other crustaceans which are being present. So these are few. Apart from that, you have ostracod larva, which are belong to a molluscan group. They are shelled ostracod species, having a very hard shell and more or less covered and lightish in very light in color. Then you have other larval forms like the jellyfish larva, which are shown in the figure. They are very tiny and they can move on their own using their tentacles, the small tentacles. Then you have your trophophore larva, which develops into your polychaetes and other what do you call it, groups in the platyhelminthes, or yeah, those platyhelminthes or annelids families. And then you have few other larval groups uh, which develops to similar, this protozoan and metazoan species. Excuse me. So yeah, these are few larvals that I can recall at the moment. So anything
Uh,
else that I would like to?
uh, no, I don't see any other questions from the students. Okay, so we'll give the vote of thanks now. So, firstly, I'll thank Mr. Abhijit for making his time and giving us a wonderful presentation on planktons in general, um, and also telling us how these amazing organisms are caught and what are their applications, their role in the environment. So, uh, so since students are all educated, I feel the students have learned much from the topics. So, thank you, Mr. Abhijit. And I'll also like to thank St. Joseph's College for School of Life Sciences, Department of Zoology, Natural Science Association for conducting this webinar uh, in the hopes of educating the students as well. So thank you guys. We'll conclude the meeting here.
Yes, even I would like to thank Mr. Chetan and St. Joseph College for giving me this chance to speak today. And for the students, in case you are not clear or you had doubt, or if I was too fast for you to understand, you can anytime contact me, or you mean in my mail or something. Then I can clarify certain doubts if you are having. Otherwise, if I was able to make you understand, then thank you for your time.
Okay. Thank you guys. I will meet again next week for in the next webinar.